Lost in translation? I hope I can help. So translation is the process of putting together amino acids or protein letters to form a protein. And today I want to tell you about how it works and how I like to think about it in terms of a lot of weddings. So proteins are kind of like the main workhorses in your cells, and they're made up of these letters called amino acids. And we've talked about how these different amino acids have different properties. And then when you link them together in these chains through peptide bonds, it's going to fold up based on like the sequence of those amino acids and their various properties. And then kind of like how a spoon and a fork will do different things, the shape that the proteins take is going to help influence their function. And so it's really important that cells be able to put together amino acids in distinct orders, so that you can get things, you can make your fork versus your spoon versus your knife. And so different proteins, have different sequences of amino acids, and their instructions for making them are held in their genetic recipes. And so you have like the permanent original recipe. So that'd be like the DNA gene. And then your cells make messenger RNA copies of that gene. So kind of like Xeroxing the gene, making a little edits to remove some regulatory information called introns. But the basic idea is you can make lots of RNA copies, lots of messenger RNA copies of a gene, and then you can make lots of protein from those. And so this in the protein making process is called translation. And in this process of translation, what happens is that this protein making complex we'll look at called the ribosome, it's going to travel along the messenger RNA. It's going to read the instructions in that messenger RNA that tell it what order to link the amino acids in, and then it's going to help link them up to form your protein. So how this works is that within the messenger RNA, the protein letters are spelled out kind of in these things called codons. So basically 3 RNA letters spell one protein letter. And how it works is that you have this go-between called a transfer RNA or a tRNA. And so on one end, it has the complementary anticodon. So this is base pairing, just like we see between strands of DNA and between strands of like RNA. You can have base pairing between, this is like the complementary, like G to C, A to T, or A to U in the case of RNA. And so when you have the complementary anticodon on this transfer RNA, it'll bind to the codon. And the cool thing about this is that the tRNA that has the anticodon that matches that codon is going to be attached to the amino acid that corresponds to that codon. So in order for all of this to work, we need this transfer RNA to get loaded with the right amino acid. And this is going to be done with the help of amino acyl tRNA synthetases we'll look at, AARSs. And then the ribosome needs to be able to help facilitate the actual linking of this new incoming peptide, this new incoming amino acid, incorporate it into the growing chain. And then this is going to go on until you reach a codon that instead of spelling a protein letter, it says stop, and then this protein called a release factor is going to come and bind and cut off the chain. And so let's look in more detail at how this is actually going to happen. So this is the more like realistic view of how things are going to go down. Then I'll show you a way I like to think about it in terms of like a translation as being like a wedding, to help understand what is going on and remember things. So the basic idea is that this ribosome has a lot of parts, but it has these 2 main like halves, but it has these 3 like sites in it where tRNA can be located, the E, the P, and the A. So it's going to enter in the A, so you can think A arrival, and it's going to exit the E, so E exit, and then the P is where the growing peptide is going to be held, so P for peptide. And so basically what's going to happen is that when the ribosome's traveling along, the tRNA is going to come in with the help of like elongation factors. When the matching one's going to bind, a new peptide bond is going to form with the growing chain transferred to the new tRNA. Then you're going to have another elongation factor come and help shift things over to the P site. So you're opening back up the A site. Things are moving along, and now you have a new codon show up in this A site. And so you can have another tRNA bind with that corresponding anticodon and with that corresponding amino acid. So that's the basic overview. And now I'm going to show you how these steps work out in more detail, as well as the way I like to think about it. So we can think of this ribosome as being kind of like a drive-through chapel with 3 parking spots. And so these would be our E, P, and A sites. And our messenger RNA is like kind of like the road that this drive-through chapel is kind of like traveling along. And so these parking sites are going to be occupied. The things that can park in it are your tRNAs. And so your tRNAs are kind of like limos, and their license plates are kind of like the anticodon. And so basically you can think of these parking spots as being reserved for specific limos. And so these are corresponding, these anticodon license plates can only park if they have that spot reserved for them by the corresponding codon. And then we're going to have elongation factors kind of be like our bouncers, helping move things along. And then this is all going to happen until you reach the stop codon. Someone comes in and is like, hey, we got to stop this wedding. There's another function going that's scheduled now, and then this will get cut off. And the newlywed protein can go and on its honeymoon or whatever, and whatever function it needs to do. So let's look in a little more detail. So let's start with our chapel, our ribosome. So it has these 2 subunits. It has this large subunit and the small subunit. And so this large subunit, it's in humans, it's the 60S, and bacteria, it's 50S. And then the small is the 40S in humans and 30S in bacteria. What these numbers refer to is this thing called like a Feldberg unit. So basically, when you can separate like complexes. So this ribosome is like a complex of protein and RNA. It's like this super complex thing, and the RNA is actually doing most of the work, which is really cool. But anyway, you can like separate these complexes or complexes of all sorts of complexes using like density gradient centrifugation. So basically you spin things really fast in like a thing of sugar or whatever, and then the heavier things are going to travel, sink faster. And so then you get information about their size. And so, yeah, so the units like are kind of weird because of this Feldberg thing. So it's not like directly proportional to, like, you can't say, oh, this one is like this many times bigger or whatever. But it gives you an idea of the relative sizes. And so you can think of the small subunit and large subunit as kind of being like 2 unequally sized prefab halves. And these are made up of RNA and protein pieces. So we're going to have our tRNA limos are going to be taking our amino acids to the chapel. And so different amino acids are going to have different designated limos. And so you're going to have different tRNAs with different 3-letter anticodon license plates. And so then they can only park in where they have a parking spot, so where they have the corresponding codon. So you can look at this thing like a codon table, and what you'll see is that some of the amino acids have multiple tRNA, they have multiple codons, so they have like multiple limos, but a single limo will only ever have 1 amino acid. So there's degeneracy in terms that multiple codon can spell the same amino acid, but there's no ambiguity. So 1 amino acid will only ever spell 1 amino acid. And they're non-overlapping. With the correct open reading frame or whatever, like telling them where to start, is going to be signified by the start codon, AUG, which if it occurs elsewhere in the protein, like after the start codon, then it'll be then it'll spell methionine. And then you also have these like 3 stop codons, which are where the release factor will bind, as we'll see. If we look at what needs to happen for a peptide bond to form, basically you need to combine the carboxyl group of one amino acid with the amino group of another amino acid. If you were to like do this in the lab or like see it in a textbook or whatever, you can see that it's like this condensation reaction overall in that you're losing a water. But this isn't how it actually happens in our body. And I have much more on this in another post in another video. I kind of like draw out every step of this process. I'm not going to go into it in detail here, but the important thing is that this is actually going to happen stepwise, and it's not going to happen just like this. Instead, we're going to be transferring the amino acid. We're going to be transferring the incoming amino acid first to the tRNA, and then from that tRNA onto the growing chain. And that growing chain itself is going to be held on by the tRNA that's in that P site. So we're going to be transferring a free amino acid to the tRNA, then from that tRNA to this chain of amino acids that are held on the tRNA that had come in before. So you want to make sure that when the ribosome's like making a protein, it's making it right, it's sticking everything in the right order. There's a bunch of proofreading steps in the protein making process. What the ribosome is going to be looking at is it's going to be looking at this, it's going to be looking at the anticodon-codon match. So basically what parking spots are reserved, and whether they have the right license plate to park there. And so you're going to have like affinity between this codon and the anticodon based on their base pairing. But you need to make sure that you have the right amino acid loaded onto that tRNA, or else you're going to add the wrong thing, even though you're using the right tRNA. So the enzyme that's going to add this amino acid onto this tRNA, we call this like tRNA charging, it's going to be need to be able to correctly bind to the right amino acid and to the right tRNA. So this is going to be done with the help of an amino acid tRNA synthetase, or an AARS, that's going to be able to recognize both of them and help facilitate this process. And so to recognize the right amino acid, it kind of has like pockets in it. So it's like so this big protein enzyme, and it has sites in it that can kind of recognize the shape of the amino acid. But because some of them are really similar or shaped and stuff, we need to make sure that we have further confirmation that we have the right one. So this is actually going to be helped out by the tRNA binding. We're going to have another like proofreading step. So in addition to like being able to recognize the right tRNA, like binding to the right tRNA is actually going to help make sure that you have the right amino acid. It's pretty cool. What's going to happen is that, so this AARS is going to recognize the tRNA. These various ones do it in different ways. Some recognize the anticodon, some recognize things in like the rest of the tRNA. Some a combination of both. But what's going to happen is that when it binds the tRNA, it's going to push the amino acid into kind of like another proofreading site on the enzyme, which is going to help make sure that you have the right thing loaded. And then once you have this loaded, now you just need to focus on that anticodon matching so that the ribosome can focus on not needing to be able to recognize what's up here, just what's down here. So in our limo analogy, we're basically putting the right amino acid on the right limo. And we're going to have to, so you can think of this as the amino acid kind of like going to the ATM to pick up a charge to pay for the ride. And so basically, this is going to require the use of ATP. So ATP is kind of like energy money. So it has these phosphate groups which are negatively charged. It's kind of like a clamped spring. These are going to be held tightly together. And so it's kind of like you're clamping a spring, which doesn't want to be clamped. So if you break one of these bonds, then you release energy. So we call these like high energy bonds because they have high potential energy. And so when we break one of these bonds, we get energy. We can make like not so favorable reactions more favorable. And what we can do is that when we when we're doing this, we're making it easier for us to do the next step. You can also think of it as like being worth at spending some energy, because you want to make sure that you have the right tRNA you want to make sure you have the right amino acids on the right tRNA. So when we're doing this, when we're adding this AMP. So basically the part, so here we're going to ADP, but so it's adenosine diphosphate, but if you take this, if you break it here instead, you're left with AMP and adenosine monophosphate, as well as pyrophosphate. So these two phosphate groups, I can then get broken up to give you even more energy. This tRNA, it has this hydroxyl group, this OH group. So this can act as like a nucleophile and like attack, much more nucleophiles and electrophiles than another post. I'm not going to get into it here, but basically this can be reactive, but it's going to want to it's not going to want to react to this. It will react to this. And so this is going to be kind of like this is going to make this easier to add to and kick this off. So this is going to be like a substitution reaction. So you want to have a good leaving group. So carbon can only form 4 bonds. If you try to form another bond to it, you're going to have to kick something else off. And so that kicks something off. The thing you kick off is your leaving group. And you want to have a good leaving group in order for this to be favorable reaction. It's a lot more favorable to kick off this AMP than it would be to kick off just like this hydroxyl. And so by adding this AMP, we are then making this more favorable to get added on. And then once this is added on, we can, with the help of the ribosome, we can make it a favorable reaction to have this react with the chain. And if you're wondering it, so like amino acyl, so an acyl is basically just like a carbonyl, so a C double bond O attached to something else. So when you have this, you're acylated. You have this as acylated. And then we call this like charged tRNA. See, I'm not going to get too much into the initiation because it differs a lot between like eukaryotes, like so plants and animals, and then like bacteria. So prokaryotes like bacteria, but it's a lot easier, better understood with bacteria. But the basic idea is that you have like, you need to get this party started, you need to have special like initiation factors that are going to help escort the initial amino acid. And so in bacteria, the initial amino acid is bromomethionine, so it's like a modified form of methionine that's going to help when the start codon comes. It's going to all help this all get set up. It's going to be a little different in eukaryotes, and I'm not going to go into that. But so now let's imagine that we have this set up. So we have this set up in the way that we have the first amino acid is going to be in our P site. And when we continue on, this P site is going to be the site where the growing chain is going to be sitting. You can kind of think of it as going to be coming out of like the chapel's chimney. But the new one's going to be coming in this A site, and then the chain is going to get transferred from this P site. This growing chain is going to be transferred from this older tRNA to the newer one. But in order to bring in the newer one, you need to have, you need an escort. And so we just charged our tRNA, so we have our loaded limo. And then we're going to have this chaperone escort come, and this chaperone is going to be an elongation factor. And these elongation factors, so in bacteria, it's like EF2, and then humans, so it's like EF1. But the basic idea is that this is going to be a GTPase. So we talk about how ATP could store energy. The same idea with GTP, except you have a different nucleotide. They have the same like triphosphate, so they could act as energies. You want to again spend some energy because it's important that you're making sure that you have the right you have the right tRNA come. And then once it sees that everything's okay, it's going to basically pay for the limo's parking by cashing in its GDP. So it splits that GTP to make GDP. So it in a free phosphate group, and then it splits, so it leaves. And when it does this, it's going to like change the change shape, and this is going to help it leave. So in the GTP form, it's more strongly binding, and then once it sees that everything's okay, then it can basically spend that GTP and then like change shape, so it no longer wants to bind as much, and then it'll leave. And this is going to help make sure that we have our specificity. But remember, this is all taking place in the ribosome. And so once that chaperone leaves, now it's the ribosome's job to help facilitate the transfer of the growing chain onto the incomed peptide that just arrived. So you're going to actually have this amino acid is actually going to kind of like nucleophilically attack this carbonyl carbon, push that off, then to attack this, push this off. So much more on how in the other post I was talking about before. But the basic idea is that now you're going to have your chain added to this, the newly arrived amino acid. And so now you might see a problem. Well, everything's in the A site, and we talked about how we want the growing chain in the P site, remember? And we need to open up this A site so a new limo can come, so a new tRNA can come. So when you have this added on, so we're going to need to shift things over to the P site, not just because we need to open up the A site, and not just because the P site is where we say things should be, but because it's going to be really awkward having it like this, having it in this state where you have this bound, but in the A state, because it's kind of like, well, you want it to go out the chimney, but now you kind of like, and these tRNA, these codon anticodon matches are making it so that these are stuck in place, but this part can kind of move more, and so it's really awkward. You have this awkward intermediate step. So now we're going to have our bouncer come in. So it's another elongation factor, EFG or EF2. It's going to come in and get them to basically move over a spot. And so once again, we're going to kind of see the spending of GTP. So we can get kind of a conformational change, or kind of like shape change, so that it'll encourage us to move over, and the GDP bound form to leave. Now you have everything shifted over to this P site, and now what was the previously there tRNA, so this guy is going to get kicked off. It's going to get kicked into this E site, and then it can leave. So this is going to keep going and keep going, but ultimately the wedding needs to come to an end, and this is going to be signaled by a termination, and by a release factor that's going to show up when you have a stop codon. So when your spot is reserved by for UAA, UAG, or GA. And in this case, you don't actually have a tRNA that's going to match those. Instead, what's going to happen is that a protein called a release factor is going to come, and it's going to help with water attack. So basically hydrolytic cleavage. And so this release factor protein is going to come bind when that shows up in the stop codon, and then it's going to cut this. This is going to then fly out of your chimney. And so overall, you have something like this, and then you make this long peptide chain that can fold up to make a pretty protein. Although it's also like it's folding as it's coming out. And so sometimes, because a part of the protein might come out like over here, that needs to be folded up with like something over here. So there's also like chaperones, these like proteins that can help kind of shield the part that's coming out, so it doesn't like misfold before the rest of it comes out. So a bunch of cool stuff on that.